in some populations, the prevalence of type 2 diabetes can be as high as 50% in the population.
Wow.
They wouldn't live that long. They would die from effectively extended diabetic ketoacidosis. There have been very large-scale studies of both albuminuria and EGFR in the general population, and they've never reported that this same genetic variant is associated with either of those traits, suggesting that it's a diabetes-specific phenomena. Initially it was thousands of dollars. Now it's probably around $20. They actually made the raw genetic data publicly available, which is actually something that would be unheard of now. SNP in the collagen 4A3 variant that was associated with reduced risk for diabetic kidney disease was associated also with differences in this glomerular basement membrane thickness in people with type 1 diabetes. And to me, this was kind of a fascinating connection between an association with effectively the amount of albumin in the urine, but also showing a connection to a very concrete histological feature that's measured very precisely on these renal biopsies from people with type 1 diabetes. It's actually probably the most important trial ever performed in people with type 1 diabetes. The average IQ of the participants in DCCT, I think, is about 120, whereas, of course, the average IQ in the population is around 100. And what we showed is that in that 10-year period, the situation had actually got even worse than better. That once we understand the genetic contributors to a disease, we then can start to understand the mechanisms of disease development and then rationally think about designing therapies that could be beneficial.
Dr. Paterson, welcome to the show.
Thank you.
What is diabetes?
It's formally defined as a blood sugar above some threshold. I mean, we can think of blood sugar or any other measure of glycemia as being a quantitative trait. And actually, the thresholds have changed a little bit over time in terms of where diabetes is diagnosed, and there's also a pre-diabetes threshold. This is mostly related to type 2 diabetes, and the threshold's been changed based upon at which levels having a glucose of that level is associated with the development of long-term complications of diabetes, mostly eye disease, some heart disease as well.
So these are complications.
Yeah.
Mm-hmm. Interesting. And you mentioned type 2 diabetes. What are some of the other types?
So formally, we can classify diabetes into kind of monogenic forms of diabetes that are typically very rare. Some are neonatal onset forms of diabetes. Some are simple Mendelian forms of diabetes that used to be called like maturity diabetes onset of the young. We now call them monogenic because their onset can be at any age, and actually kind of putting the word young into the title of them kind of potentially can be misleading. But and there are a number of Mendelian diseases in which diabetes is more common than in the general population. So, for example, cystic fibrosis. People with cystic fibrosis can develop diabetes or do develop diabetes at a higher risk than the general population. So there's a cystic fibrosis-related diabetes. There are other Mendelian diseases in which diabetes features as a part of the component. But the main two classes of diabetes are type 1 diabetes and type 2 diabetes.
And interesting. But that sounds very interesting because it feels like it covers both monogenic and polygenic types, which I think it makes it very interesting to study. But I mean, how different is, I mean, like given the range of all of these types, I mean, they are all formally named diabetes, but like how different are they? Can we like actually think of them as just one disease, or they're so different that, you know, it's not really a good idea to classify all of them as diabetes. Like we can think of cancer as one disease, but we have many types and forms and cases that it's just like each one has its own field.
Yeah. I mean, originally diabetes was thought of as a single concept, but it would realize that mostly based upon age of diagnosis, there's at least two forms of diabetes. Type 1 diabetes, which typically occurs relatively young in life, although we're starting to understand that that's not necessarily the case. And at least prior to the discovery of insulin and our ability to synthesize it and deliver it, it was effectively a fatal disease because people with type 1 diabetes effectively produce little or no insulin themselves, and they require exogenous insulin to survive.
And sorry to interrupt, so they would just die because of the complications like kidney failure and...
No, not of long-term complications, because they wouldn't live that long. They would die from effectively extended diabetic ketoacidosis.
Oh, I see.
Yeah. And before insulin was available, if you read back kind of the stories of some of the first patients to receive insulin, effectively they'd been living for months on this very low calorie diet, kind of usually low carbohydrate diet, trying to preserve their life using dietary methods. I mean, it did extend their life to some extent, but it was at very low quality and not for a very long period of time.
How long ago is this?
So insulin discovery, I think, was 1911, but I could be in error. There was some celebration recently, so it actually maybe slightly after then.
Right. So if you want to map out, like if we map out all the diseases along a continuum where one end is monogenic and the other end is polygenic, where would you place each type of diabetes?
So the monogenic forms of diabetes are clearly monogenic. Type 1 diabetes is relatively simple from the perspective of complexity. So it's closer to monogenic than something like type 2 diabetes. And the reason that type 1 diabetes is more simple is that there's a very strong effect of specific HLA haplotypes in the major histocompatibility complex on chromosome 6, impacting the risk of developing type 1 diabetes. And that was discovered, I think, in the 1980s using relatively small sample sizes and using serology for HLA typing.
And well, since you work at SickKids, which is a children's hospital, how common is diabetes among children? Because whenever someone says diabetes, I just picture my grandpa, like, someone old. And it feels like cancer, like a disease that you develop when you're like quite old and as a result of aging, I guess.
So the different forms of diabetes have different incidences in different populations. So people of European ancestry, the lifetime risk of developing type 1 diabetes is probably around 1 in 300. In non-European ancestries, it's typically lower. In some cases, probably 10 times lower. And that's mostly due to differences in the frequency of these HLA haplotypes that are major risk factors for type 1 diabetes. They happen to be higher in people of European ancestry than in people of non-European ancestry. For whatever reasons, it could just be because of genetic drift and how genetic variation has changed in frequency over time. Type 2 diabetes, the other major form, it's a disease that has generally been increasing in incidence over the last half century, I would guess.
Why?
For which we have good data. Mostly it's thought to be related to industrialization, people doing less activity, eating different types of food, and not exercising as much as they probably should do. In some populations, the prevalence of type 2 diabetes can be as high as 50% in the population.
Wow.
And again, there are ancestry differences in terms of the cumulative incidence. In fact, European people typically have some of the lower incidences of type 2 diabetes. People from indigenous North American populations have some of the highest risk. People of Black African ancestry, their risk is increased compared to Europeans. People from South Asia, and also some East Asian populations also have higher risk than people of European ancestry.
So all these like different statistics, do they also match with the patients that you see in real life?
Yeah, like at SickKids Hospital, although the GTA is a very multicultural region, still probably the majority of patients with type 1 diabetes seen at SickKids are people of European ancestry. Whereas in a type 2 diabetes clinic, I'm not familiar with them, but one would imagine that there's a overrepresentation of people of predominantly non-European ancestry.
So as a clinician, of all the diseases that you could study, why did you decide to study diabetes?
I think it started off when I was training and people were starting to do genetic studies of common complex diseases. If we go back actually a little bit before that, people were starting to successfully identify the genes that cause rare Mendelian diseases. Like the classic example was like Huntington's disease, and then cystic fibrosis. And then people, or the research community, wondered whether we could use some of these general strategies to identify genetic variants that impact the risk for developing more common diseases. So some of the earliest genetic studies of common diseases were of type 1 diabetes. Effectively, they had recruited families where two siblings were both affected with type 1 diabetes. And genetically, they were looking for regions of the genome where those two affected siblings were genetically more similar than we would expect by chance based on Mendel's laws. And that actually supported the evidence that there's a major locus at this MHC region, the HLA genes, and identified some other regions in the genome as well. The problem with that strategy, which is what we generally would have called affected SIB pair linkage analysis, is that it's relatively low-powered to detect modest genetic effects. The effects need to be quite big to be able to be identified using that strategy. But in fact, the people who were doing the first genetic studies of type 1 diabetes, they actually made the raw genetic data publicly available, which is actually something that would be unheard of now because of potential identifiability of the subjects. But that allowed myself and others to be able to use that data or examine that data for various phenomena, perform additional analysis on it. So I kind of became very interested in that data from that time.
I think we made a great case for studying diabetes in the first place. But the next question would be, why do you study it through the lens of genetics?
So I've always been fascinated about trying to understand the fundamental causes of a disease or of a trait in the population. And it's clear that people with diabetes have increased risk for developing long-term complications of diabetes, so eye disease, kidney disease, heart disease, etc. And in some populations, they have reduced life expectancy as well. So if we could understand the causes of the disease, we could either prevent the disease or treat the disease in a more informed way that would reduce the risk of people developing the long-term complications of the disease. So those were the main motivations of why I wanted to try and understand the causes. And we'd learned from simple genetic diseases like Huntington's disease or cystic fibrosis that once we understand the genetic contributors to a disease, we then can start to understand the mechanisms of disease development and then rationally think about designing therapies that could be beneficial, as opposed to kind of without the genetic knowledge, effectively trying therapies at random without a clear understanding of the mechanistic causal pathways. And the problem of doing effectively everything else apart from genetics is that we can easily get confounded or misled by confounding. Like we can see an association between some particular measure that we've obtained from the blood and a disease, but we don't know whether that's causally associated with the disease or whether actually the disease causes that abnormality. So genetics allows us to understand the causal pathway to the development of disease. So to me, that's kind of a very powerful strategy.
Interesting. And you mentioned association. So I want to ask you this. When you want to link genetics to the phenotype or the disease, what are the tools in your arsenal that you use to go from genetics to the disease and phenotype?
So since the completion of the Human Genome Project, we started to get very good catalogs of the genetic variation in the human genome. The human genome is large. There's about 3 billion nucleotides in the human genome. And the current database of genetic variation in the human genome, called dbSNP, has about 1 billion SNPs in it. So that means that on average, 1 in 3 nucleotides in the human genome has been seen to be polymorphic in at least one individual from it's actually millions of people that have been sequenced so far.
Oh.
The other thing that was realized soon after the Human Genome Project was completed and we had a draft sequence of the human genome is that genetic variations that are close to each other tend to be correlated in the population. So even if on average there's millions of variations between two individuals, if I wanted to capture the majority of those, I don't need to measure all of those variations. I could measure a subset of them and take advantage of the fact that the nearby genetic variations are correlated with each other in the population. A phenomena that we formally called linkage disequilibrium, but effectively it's really just local correlation. So that led a number of companies, along with advances in technologies, the ability to genotype these genetic variations that we call single nucleotide polymorphisms, or SNPs, SNPs, allowed them to develop technologies where we could place about 1 million SNPs on an array and measure them simultaneously in sets of individuals. And these became available to the research community and actually are now available even to the general public through direct-to-consumer genetic testing companies like 23andMe and Ancestry.com, where at scale people can measure about 1 million SNPs for, the cost in very large studies is probably less than $20 per sample in a research setting. So very low cost to measure a lot of variation. And
so $20 now or back then?
Oh, initially it was thousands of dollars.
Okay.
Now it's probably around $20. But relatively cheap ways of measuring the vast majority of genetic variation in tens, hundreds, thousands, tens of thousands, millions of individuals at relatively low cost. And that's really what's driven the vast majority of large-scale genetic studies. Now that we have next-generation sequencing, specifically, say, whole genome sequencing, which allows us to effectively catalog all of the variation in each individual, people are starting to do whole genome sequencing genetic studies. but it's still relatively expensive compared to using these GWAS arrays.
Mm-hmm. So this gives you genotypes.
That's right.
Right. And how do you go from that genotype to the phenotype? You do some sort of like statistical testing?
Yep. We clean the data, first of all. So often there may be some sample mix-ups or things like that. So we will compare, say, individual sex chromosome composition from the genotype data, like the numbers of copies of the X and the Y chromosomes, to their self-reported sex. We'll sometimes compare their genetically estimated ancestry to their self-reported ancestry. Again, a good way of picking up potential sample mix-ups. And we'll remove some SNPs that don't perform particularly well. Sometimes there are nearby SNPs that are interfering with the assay that cause either incorrect genotypes in a systematic way or missing data in a systematic way. So we'll remove SNPs like that. Sometimes DNA samples from multiple people get mixed together, and that produces unusual patterns in the genotype data. We'll try and identify those and throw them out as well. But then once we have clean genotype data, effectively our statistical analysis is usually some kind of regression setting where we test the 3 possible genotypes at a SNP. Because humans are diploid, we have 2 copies of each allele. We test that the genotypes of the SNP for association with a trait. The trait is usually either dichotomous, a disease, or control, or kind of dark hair, light hair, hair, no hair, these kind of phenotypes, or a continuous trait like blood pressure or height or cholesterol levels or anything we can think of.
Mm-hmm. So for your research specifically, I mean, when you look back at all the studies that you've done, where did you usually get your genotypes from, and what phenotypes did you use?
So the genotypes come from mostly patients in either cohort studies or from clinical trials. Usually we use DNA from blood just because that's what's being collected and stored, although like the direct-to-consumer genetic testing companies use DNA from saliva. There's usually not much difference in the quality.
Really?
Yeah.
So interesting.
So although I mean, depending upon how long it is since you last ate, there'll be DNA from the things that you ate in your mouth, but there's enough DNA from your cells that have been sloughed off from the inside of your mouth. The other option is using some kind of cheek swab, effectively just a swab that you kind of rub against the cheek to collect DNA. And because the assay is specifically measuring or designed to measure variation in the human genome, it's not going to pick up or be interfered with by variation or DNA from non-human sources.
So the fact that most GWAS studies like still use blood is just because some other phenotypes that they want to get out of blood, or I mean, do they even use blood anymore? Like
most studies would collect blood and measure various things as well as store blood for the purpose of measuring things that would in the future become available, novel biomarkers. And often, like I would guess from probably like the 1990s onwards, most large-scale studies, especially pharmaceutical studies, would have collected blood, extracted DNA from an aliquot of it and stored it for future use. Yeah.
Interesting, because I've always looked down on like all the studies that they've like used saliva to collect DNA, and I was just like brush them off as low quality. But
I mean, 23andMe, as I understand, have like 15 million customers that have all provided saliva DNA.
Yeah, I'm one of actually.
Yeah, so am I. So there's nothing wrong with saliva.
So back to the phenotypes. For diabetes, specific phenotypes, I mean, one phenotype obviously would be you either have diabetes or don't.
Yeah.
What about like other phenotypes that you've used in your work?
So one of the studies that we've done a lot of genetic work on is individuals that were enrolled in a randomized clinical trial called the DCCT trial, stands for Diabetes Control and Complications Trial. It's actually probably the most important trial ever performed in people with type 1 diabetes. And the goal of the trial was to answer the question, if you could make people with type 1 diabetes effectively normoglycemic, if you could push their glucose level back into the normal range, could you delay the development of long-term complications of diabetes and prevent the initiation of some of these complications? So the trial was run in North America. There are actually sites in Toronto and in London, Ontario as well. And roughly 1,400 individuals were recruited and were randomized to the two treatments. The conventional therapy that at that time was effectively trying to maintain people with type 1 diabetes in a symptom-free manner, so effectively not allowing them to become extremely hyperglycemic, but also not hypoglycemic. But that was mostly done by testing urinary glucose at that time, which really is a test that in most developed countries is no longer used because there's this renal threshold for glucose. So only when your glucose goes above a certain level is glucose pouring into the urine. So that was the conventional arm, effectively trying to keep them relatively stable, similar to what conventional therapy was in the community at that point, versus intensive therapy, where the goal was to make them biochemically normoglycemic. And that was achieved either by multiple daily injections of insulin or continuous infusion of insulin with frequent blood glucose monitoring. I think at least 4 times a day, but kind of typically before and after every meal. And although the goal was to make them normoglycemic, because of the technology that was available at that time, it was not technically feasible to do that. But certainly their average glucose levels and their hemoglobin A1C levels were much closer to the normal range than they were to the people assigned to conventional therapy.
Mhm.
So getting back to the question of what traits did we use, we effectively could analyze any trait that was collected in this trial. So there were lots of traits measured at the baseline of the trial to ensure eligibility for the trial. So, for example, one is C-Peptide. The C-Peptide is a byproduct of insulin secretion. So although we think of type 1 diabetes as people who effectively are not secreting any insulin, when we measure C-Peptide, and the reason we measure C-Peptide in people with type 1 diabetes is because C-Peptide has a longer half-life than insulin does, so it stays around in the body for longer, partly related to its first-pass metabolism in the liver. But also, the assays at that point couldn't distinguish between endogenous insulin that the person's producing themselves and exogenous insulin that they were injecting. So the field still uses C-Peptide as a measure of beta cell function or insulin secretion in people with type 1 diabetes. So we actually published a follow-up paper just earlier this year where we can see in these people with type 1 diabetes who classically we would think to have no C-Peptide, they actually have a range of C-Peptides, usually much lower than the normal range in the population, but kind of at least a third of them had detectable levels even using the assays that were developed at that time. So we have previously and most recently analyzed the level of C-Peptide at baseline of the trial as one of the traits that we're interested in, trying to understand the genetic basis of differences within people with type 1 diabetes, because people who have residual C-Peptide typically have better glycemic control over their life with diabetes. They require less insulin because they're actually producing insulin themselves. And they have lower risk of developing long-term complications of diabetes as well.
Is that something purely genetically driven, the C-peptide levels?
It has a genetic component to it, like all of the traits that we're studying. I wouldn't say that it's purely genetically driven. The evidence that we have at the moment is it's about a quarter of its variance in people with type 1 diabetes is genetically influenced, although those estimates are relatively imprecise. And we really have very little information about what the non-genetic factors are that are influencing the residual C-Peptide secretion in people with type 1 diabetes.
And so this question might not be like directly related to our conversation, but when you learn that some phenotype like C-Peptide is not purely genetically driven, would you consider that as good news or bad news? Because if it's all genetics, maybe we can change that. But if it's environment, maybe we can, like, it's so easier to change the environment perhaps. Like, would you rather the phenotype to be monogenic or polygenic?
I don't think of phenotypes, like very few phenotypes that we work with in people with diabetes are monogenic. In general, we used to have to justify why we wanted to study a particular trait from a genetic perspective. And we would typically rely upon classic studies like family studies showing that the trait is correlated in families, twin studies that the trait shows higher correlation in identical twins than in non-identical twins.
This is before GWAS?
Before GWAS. But now, especially in studies like the DCCT study, where we already have the genetic data, we have hundreds or thousands of traits measured on these people. Like, there's not much of a barrier to us doing a GWAS of a particular trait. And I wouldn't necessarily choose traits upon ones that either I thought had big genetic or small genetic influences on them. More, I'm just interested in identifying some of the genetic influences on the trait, irrespective of what prior data may be available.
Right. I love the point that you mentioned about like these biobanks basically enabling GWAS, because as you said, maybe if we didn't already have the data, we had to like write grants and convince people that like it's a good use of your money if you just like do the genetic stuff. But I mean, we already have the data, so why not?
Yeah, so like in the UK Biobank, where there's about 500,000 people who've already been genotyped, some groups have done effectively GWAS of every trait that's ever been measured or collected on these people. So they have GWASs of around 4,000 or 5,000 traits. Similarly, in the Million Veterans Program, another large biobank where about 600,000 people have GWAS data, they've recently published a paper where they did GWAS of something like 4,000 diseases and traits, mostly pulled from electronic medical records and administrative billing information as well.
Amazing. So now that we have a good understanding of diabetes and GWAS, let's talk about some of your major discoveries. And if you go back in time, in 2010, you found the first genetic locus that affects blood sugar control within people who have diabetes. And this wasn't really about getting diabetes in the first place, but controlling it. So can you tell us the story of the SORCEUS1 gene discovery?
So that genetic association came from our strategy where we were selecting traits from the DCCT participants for genetic analysis. And in fact, one of the goals of intensive therapy that I mentioned earlier was to try and make people with intensive therapy get their A1C below the diabetic range, so below 6.5%. And I think the intensive group, they were in the trial for an average of 6.5 years. They had hemoglobin A1C measured every month for 6.5 years. The conventional group, I think, had A1C measured every 3 months, again, for the same average time. So we have this immense amount of longitudinal data on these individuals for a quantitative trait. And other groups had started analyzing various aspects of the genetics of glycemia. there'd been some large-scale studies of fasting glucose in the general population, and also some genetic studies of hemoglobin A1C in the general population, because also at that time there was a shift in most diabetes societies in terms of the recommendations for screening for diabetes. Prior to that, people were mostly screening using a fasting glucose or a 2-hour glucose after an oral glucose tolerance test. For various reasons, most organizations shifted to using hemoglobin A1C as the initial screen for diabetes in the population. So we were interested in the genetic aspects of hemoglobin A1C. So what we did in that paper was we took the two treatment groups, because their A1Cs were drastically different after the start of the trial. And we did GWAS of initially just their average A1C during the trial, separately by the intensive group and the conventional group. And then we also did an analysis that combined the two groups together, and we looked for differences in the genetic effect sizes or the SNP associations based upon the treatment group assignment as well. So we identified a number of signals, of which this SNP near this gene SORCS1 was the most significant association. And once we'd identified the association using the average over time, we then also did some more sophisticated statistical models where we took advantage of all of the repeated measures that each person had over time. Especially when there's a lot of variation within a person over time, that potentially can be more powerful than just kind of taking a simple summary measure like the mean.
Mm-hmm.
The interesting fact from that study is that although we saw a strong effect, that genetic association has, as far as I'm aware, never been reported in people without diabetes, suggesting that it may be a diabetes-specific phenomena. And I'm not aware that any other study has replicated that association. That may be because, in general, people with type 1 diabetes don't get hemoglobin A1C measured very frequently. And also, because the DCCT trial, the effective intensive therapy was so marked, effectively it delayed the development of new complications and slowed the progression of existing complications. Intensive therapy has become the standard of care for people with type 1 diabetes. So most people with type 1 diabetes are now following some form of intensive therapy, effectively trying to make themselves effectively non-diabetic from a glycemic point of view. And there's less variation in glycemia in a contemporary type 1 diabetes population than we would have seen during the trial.
Mm-hmm. Were you surprised to see a signal, or were you expecting something to pop up?
Because there'd been some genetic studies of hemoglobin A1C in the general population, we thought we would see some signals. We did replicate some of those signals already, but we was a bit surprised to see such a strong signal. I mean, the other consideration is that the people that were recruited and selected into the trial, into the DCCT trial, were not a random sample of people with type 1 diabetes. So around 1,400 people were included in the trial, but they screened over 7,000 people to get those 1,400. And the screening process was actually very complicated, included various neuropsychological assessments to determine whether the participant, if they were randomized to intensive therapy, would likely be able to complete the protocol, because it required kind of a lot of attention to detail and knowledge about kind of the effects of insulin and glucose monitoring and these kind of things. So just as an illustration of how non-representative the cohort is, the average IQ of the participants in DCCT, I think, is about 120. Whereas, of course, the average IQ in the population is around 100. And I don't think that the IQ of people with type 1 diabetes is any different from the people without type 1 diabetes. So The study population was highly selected in various complex ways that may make them not very representative of the general population of people with type 1 diabetes.
Let's go back to the study design. You mentioned two groups, conventional and intense care.
Intensive, yeah.
Yeah. What is the difference? Like, what does each mean?
So effectively, for a person with diabetes, it indicated how often they should monitor their plasma glucose level and how they should adjust their insulin dosing based upon those measurements. In a very crude way, conventional therapy were effectively treating their diabetes blind to their glucose level. Effectively, they were giving themselves kind of standard doses of insulin that they worked out with their healthcare providers, but not really adjusting those doses based upon exercise or diet. Whereas the intensively treated group were effectively being browbeaten by their physicians, and their diabetes nurses were the kind of most involved in their care, to effectively try and keep themselves within the normal range of glucose. The challenge with that at the time was that by keeping themselves closer to the normal range, or as close to the normal range as they could, they would much more frequently become hypoglycemic, so have a low blood glucose level, which itself is a serious complication of diabetes. So it's a very delicate balance of kind of keeping themselves in the normal range, but not going too far. So the trial, I think, started in 1983 and concluded in 1993. So at that time, the technologies available for glucose monitoring were effectively you would stab yourself and you kind of use a strip. Insulin injections were either kind of single injectors or occasionally pumps, but very crude pumps. If we compare them to the contemporary setting where people with type 1 diabetes have access to continuous glucose monitors that are giving kind of effectively instantaneous glucose at every minute of the day, the technology has changed drastically, which helps avoid people with intensive therapy from having these hypoglycemic episodes.
And you did the conventional and intensive for 6 years.
That was the trial, yes. I was not involved in the running of the trial. I got involved in the genetic studies a number of years after the trial had concluded.
Interesting. So 6 years later, you had another breakthrough, but this time for diabetic kidney failure. You found this variant in the COL4A3 gene that actually protects against kidney failure. And well, I think that is a fascinating story. Can you unpack that?
So we'd been interested in genetics of other traits that had been measured in the trial. So in the DCCT trial, the primary outcome was retinopathy, so eye disease. And the trial was stopped prematurely because of beneficial effects on that outcome. But the trial also measured other things, including aspects of diabetic kidney disease, specifically the amount of albumin in the urine, albuminuria, as well as measures of kidney function. The contemporary one that we would use now would be this estimated glomerular filtration rate, or eGFR. So the trial, when it was stopped for the retinopathy outcome, actually intensive therapy also had benefits on aspects of kidney disease, so lower rates of albuminuria in the intensively treated group. So we've been interested in genetic studies of diabetic kidney disease, as had many other investigators. And at one point, there was an opportunity for a collaboration of many different type 1 diabetes cohorts to come together and work on what we call a Meta-GWAS. So it's like a meta-analysis that you would do in clinical trials, but here we're doing meta-genome-wide association studies. So each group would do the genome-wide association study themselves, and then we would provide the SNP association results to a set of analysts that would then combine the data from all of the different studies together to take advantage of the much larger sample size.
And these GWASs are on eGFR.
So that GWAS actually, I think, as I remember, had 10 different kidney function outcomes. I don't think we ever in that study analyzed continuous albuminuria because not all of the studies had measured it continuously. But most studies had kind of a categorical above or below some particular threshold of milligrams of albumin per urine per 24 hours. So the advantage of the meta-analysis was that although the DCCT study had exquisite amounts of data in terms of repeated measures of albuminuria over time, we actually had relatively small number of people who had developed diabetic kidney disease, either by an albuminuria definition or by a EGFR definition. And the sample size was only ever going to be 1,400 individuals because that's how many people were enrolled in the trial. Whereas we could take advantage of mostly observational epidemiological studies from many different places, some in the US, some in Europe. So there was a large study from Finland, a large study from Scotland, studies from Ireland, from other countries as well, put all together, such that we had nearly 20,000 individuals with type 1 diabetes that were analyzed for these 10 different measures of diabetic kidney disease. And the most striking finding to me was this association of this variant in the collagen 4A3 gene. It was associated with about probably at least half of the 10 phenotypes that we examined, did meta-genome-wide association analysis for, and had a relatively strong effect. The specific variant that had the smallest p-value is actually a missense change in the collagen 4A3 gene. So it changes one predicted amino acid to another predicted amino acid in the coding sequence of the gene. And perhaps a little surprising, that variant, it's actually the minor allele, at least in European populations, that's associated with protection from diabetic kidney disease. When we do large-scale genome-wide association studies, we actually don't care whether it's the minor allele or the major allele, and these are just defined by frequency, the minor less than 50%, the major above 50%. We don't care which allele is associated or which one has the higher risk. The statistical analysis is insensitive to that. But when we look at the effect size estimate, we can see whether it's the minor allele increasing the risk or the minor allele decreasing the risk. In this case, it was the minor allele of a relatively common SNP that was reducing the risk of diabetic kidney disease. The interesting thing about that study was that in a subset of participants that we included in that analysis, they came from a different clinical trial called the RAS clinical trial. That trial, although it was relatively small, it had about 300 individuals in it. It had some exquisite measures of kidney disease in people with type 1 diabetes. In fact, that trial was designed as a randomized trial of either an ACE inhibitor or an angiotensin receptor blocker on histological features of diabetic kidney disease. So that trial included renal biopsies and electron microscopy assessment of those biopsies, both at the baseline of the trial and then at closeout of the trial, which was 5 years later. And I'm not an expert on the renal aspects of diabetic kidney disease, but one feature of diabetic kidney disease that can be measured on electron microscopy is this glomerular basement membrane thickness. Effectively, they measure quantitatively how thick the basement membrane is in the glomerulus. And we were able to use the data in those roughly 300 individuals to show that the SNP in the collagen 4A3 variant that was associated with reduced risk for diabetic kidney disease was associated also with differences in this glomerular basement membrane thickness in people with type 1 diabetes. And to me, this was kind of a fascinating connection between an association with effectively the amount of albumin in the urine but also showing a connection to a very concrete histological feature that's measured very precisely on these renal biopsies from people with type 1 diabetes, providing potentially a mechanistic process for how this variant is impacting the kidney that then potentially has functional effects on how much albumin is leaking across the glomerulus.
Mm-hmm. And I think what's so interesting is that this, whatever that is, this mechanism only gets activated once you get diabetes. So it doesn't protect against kidney failure in general population. Is that right?
That's what we understand, yes. There have been very large-scale studies of both albuminuria and EGFR in the general population, and they've never reported that this same genetic variant is associated with either of those traits, suggesting that it's a diabetes-specific phenomena. Effectively, we can think of what we call gene-environment interaction, where in this case the environment is having hyperglycemia, although it may be only modest hyperglycemia. Effectively, that SNP effect is only occurring when the glucose is above a certain level. And we actually have some data from both the DCCT study as well as from, I think, the Finnish study in that same paper where we show that the genetic effect does differ by the level of glycemia. Whether this effect, whether this specific genetic variant influences risk in people with type 2 diabetes is still not clear. My reading of the current literature is that it doesn't, but the studies in people with type 2 diabetes tend to be drastically underpowered, especially because people with type 2 diabetes can get kidney disease from many different causes. So, for example, many people with type 2 diabetes will also have hypertension. There's hypertensive kidney disease. So that may be contributing to a lot of the kidney disease that are being seen in people with type 2 diabetes. Whereas in type 1 diabetes, two things. One is that most people don't develop hypertension, at least at relatively young ages. And even if they do, they're usually so well monitored in the population that they're given antihypertensive therapy to prevent them becoming hypertensive.
Right. So my whole PhD project revolves around the variant to function problem.
Right.
So I'm very keen on asking you this next question. How do you go from a new genetic signal from, say, GWAS to discovering new biology? Like when you find, let's say, a variant near the RORA gene affecting kidney function in diabetics, like how do you go from that piece of information to what's actually happening in the kidney?
So the first thing I would do if I discover what appears to be a novel genetic association is that I would search for that variant and or gene in something that's called the GWAS catalog. This is a database where effectively the results of all genome-wide association studies are publicly available. It started off with just the top signals or the genome-wide significant signals, but now tends to include the GWAS results, including all of the SNPs that have been tested but are not significant. So I would look to see whether there's evidence that the SNP or gene has been associated with any trait previously, because that potentially could tell us about causal mechanisms. Like we've associated it with a particular trait, but it may be associated with many other related traits, or in some cases, unrelated traits. It gives us a better understanding of the potential mechanisms by which the SNP is impacting either differences in the trait distribution or differences in disease risk. So that would be the first place that I would look. I mean, other people may have solved that question already, but the number of variants that have been identified from GWAS across thousands of traits is hundreds of thousands of variants, and the likelihood that people have clarified the causal effect of any specific variant is very unlikely. The next thing I would do probably would be to look to see whether this genetic variant is reported to be associated with the expression of genes.
Uh-huh.
And the data set that I would look at there at the moment would be something like the GTEx data set. GTEx is a study where they recruited around 1,000 individuals. I think most of them were individuals who'd recently deceased, and they obtained tissue from about 50 different tissues from each individual. They then used RNA sequencing to measure the expression of potentially every gene in these 50 tissues. And they also sequenced the DNA from these individuals. They did whole genome sequencing. And then in the same way that we can do a GWAS of, say, hemoglobin A1C in people with diabetes, they've done GWASs of the expression of every gene in every tissue, effectively. And the results of that are publicly available. We call that kind of aspect of GWAS eQTL analysis. So identifying SNPs that are associated with differences in gene expression between individuals in tissue-specific manner. So you can search the GTEx dataset by just putting in a SNP ID. It will tell you whether that SNP is associated with the expression of any gene in any tissue. It will also tell you more complicated things. So they have models in GTEx about alternative splicing. So sometimes some genes don't include all exons or include additional exons that are not in the canonical transcript. So sometimes a SNP is associated with a specific isoform of a transcript rather than kind of the most common one. And we can take that information. And then more recently, there's similar data for proteomics. So there's now been a number of large-scale, mostly plasma proteomic studies where they're measuring about 3,000 to 5,000 proteins in plasma of individuals from various cohorts. One is UK Biobank, where they have about 50,000 individuals. The Decode study from Iceland has a data set of around 30,000 individuals. So I would look to see whether the SNP is reported as being associated with differences in protein levels. So that would start to give us some clues about whether anybody else had previously identified this SNP to be associated with anything else, and specifically whether this SNP could be impacting the expression of nearby genes. And the GTEx database doesn't just tell us the nearest gene, it tells us all genes that are associated with it. So you may find, and it is quite common, that there are SNPs that are associated with the expression of multiple genes in a region. So that provides some information that kind of this SNP is impacting the expression of genes, but it doesn't necessarily narrow down the problem because you may have multiple genes whose expression are associated with the SNP. In the same way, the proteomic data may be similar as well. That SNP may not be associated just with the levels of a single protein, but potentially multiple proteins. And one of the limitations of the proteomic data is that most proteomic assays are only measuring a subset of proteins at the moment, around 3,000 to 5,000, whereas we know there are about 20,000 genes. Like there, of course, are way more proteins than 20,000, but kind of the assays are kind of predicated on usually the most common transcript or isoform of a protein. So when we don't see an association with a protein, it may be just because there's no current assay for that in the large-scale proteomic studies.
And in terms of time frame, like how long usually does it take from a GWAS to any functional follow-up?
It depends. Personally, I've not done much in terms of functional follow-up. In some of the studies that we've been involved with, kind of the studies identified tens or hundreds of genetic association signals. Sometimes, especially early on in GWAS, there was much more of an emphasis in terms of replicating those associations before going on to functional characterization. Now we would be interested in taking advantage of publicly available functional data rather than necessarily trying to generate it ourselves. Because if we wanted to generate it ourselves, let's say we saw an effect of this missense variant in the collagen gene. Kind of, we would want to somehow examine cells or tissues that had alternate forms of this amino acid and their effect on kind of effectively the structure and the function of this collagen protein. We know that this protein actually trimerizes with two other members, collagen 4A4 and 4A5. And we would also want to think about examining the structure of that complex as well. Like in a naive fashion, we could think about generating a mouse with these alternate alleles. It turns out that the collagen 4A3 variant, most mice actually have an alternate amino acid at that position. So effectively, we would need to generate two alternate mice with the two different human forms. And then if we think that the genetic effect is only expressed in the setting of diabetes, we would then need to make these mice diabetic or use a strain that's prone to diabetes, but then keep them alive for a long period of time to see whether that impacts aspects of their kidney function.
So if we like imagine a funnel where the top of the funnel is GWAS and the bottom is like a drug in new treatment, would you say you're more like at the top of the funnel generating GWAS results?
Yeah, certainly at the top of the funnel. There's been a interesting emphasis over the last few years about how important genetic data is related to the success of the drug discovery pipeline. So initially, like 20 years ago, a number of pharmaceutical companies had large-scale genetic projects. They were probably a little bit naive, and probably the only company that survived from that era is Decode, this Icelandic genetics company. But in retrospective analysis of drugs that have successfully come to market, they've shown that having a genetic association signal for the target of that drug means that that drug is about 2 to 3 times more likely to succeed to market than not having genetic evidence. So even if the specific genetic variant itself is not necessarily the target of the therapy, having the identified gene potentially can help the pharmaceutical companies develop novel therapies for disease.
And one last question on GWAS. You have been a vocal advocate for including the sex chromosomes on genetic studies, which many researchers still overlook. So I have 2 questions for you. Why do people usually prefer to skip sex chromosomes, and why do you care so much?
It's not really clear to me why people have skipped the sex chromosomes. I think a number of things contribute to it, probably. One is the data is a little bit more complicated than it is for the non-sex chromosomes. So males have 1 X, females have 2 X's. Females, effectively, their X chromosome can be treated in the same way that we treat the autosomes, the non-sex chromosomes. But males, we have a different strategy. And generally, when we've done GWAS, we've mostly analyzed males and females together. So then when we want to analyze males and females together on the X chromosome, we have to think about how we code the genotype data, how we build the models. Should we have sex as a covariate? Should we test for a interaction between sex and the genetic variant? And then the other thing that's probably discouraged people is that when we do these Meta-GWAS, usually we're combining data from many different studies. These studies have used different GWAS arrays in general because the companies have developed different arrays over time, made improvements, tried to compete against each other, such that kind of most studies are mostly being genotyped on different arrays. When we do Meta-GWAS, if we just restricted our analysis to the SNPs that were common across the different arrays, we would have very few SNPs to analyze. So what we do when we do Meta-GWAS is we first impute ungenotyped variants. And this takes advantage of this concept that I was discussing earlier, that nearby genetic variants tend to be correlated with each other in the population. So if I know your genotype of a particular variant, at a nearby genetic variant, I can have a very good guess of what your genotype is. And we can use external data sets in the current era, very large data sets that have high coverage whole genome sequence data, like the TopMed project, to impute effectively the vast majority of the unmeasured SNPs in a particular data set. Then what we do is we analyze both the imputed and the genotype SNPs for association with a trait in each study separately, and then we meta-analyze across the studies. So that means we have a consistent set of variants across all of the studies. The problem until recently is that the data sets that allow imputation on the X chromosome have not been particularly powerful. So people may have been concerned about the quality of the imputation, and therefore they're kind of being reluctant to do the imputation on the X chromosome and therefore present the X chromosome signals. Why am I passionate about it? I mean, when we run a GWAS array on an individual, we get genetic variation on the autosomes, on the sex chromosomes, and actually usually on the mitochondria as well. And since we've paid for it, we might as well use it.
Yeah.
And now the emphasis is for common diseases. We don't just want to identify your genotype at 1 or 2 loci across the genome to predict risk for disease. For example, predicting risk for, say, breast cancer in the population, there's a genetic score that combines like 100,000 SNPs across the genome in terms of getting the best performing multi-SNP predictor for breast cancer, and similarly for many other diseases. So why would we want to exclude the X chromosome from that predictor when potentially we have good quality data and there are likely signals on the X chromosome? I mean, if we go back to simple Mendelian diseases, because for what we historically would have called X-linked recessive diseases, we have this very obvious inheritance pattern. Only males are affected and they inherit the disease from carrier females. Yes. So it's very easy for a Mendelian disease to have a good idea of where the disease gene was located if you had that kind of inheritance pattern in a pedigree. For common diseases, and in fact, there's way more Mendelian disease genes on the X chromosome than on the autosomes, perhaps partly because it has this very simple pattern of inheritance that effectively most people could recognize simply. So although there may be, for complex disease, there may be slightly fewer signals on the X chromosome than on the autosomes, we know that for most diseases, the best genetic predictors are combinations of hundreds of thousands of SNPs. So why would we want to exclude the X chromosome from that calculation? Kind of doesn't make any sense to me.
Do you think there's been any improvement since you published that paper?
So actually, the paper we published was a follow-up of a paper that was published 10 years ago that said that there was a problem with the X chromosome. And what we showed is that in that 10-year period, the situation had actually got even worse than better. So I haven't looked recently at the data, but every time I go to a talk and people are showing me a Manhattan plot, which is this figure that describes the genetic association for a trait across all of the chromosomes, if I see that they have not analyzed the X chromosome, I may discreetly ask them why not.
I think the only like practical solution is that if all the top journals like they put a mandate like unless you have 6 chromosomes, we're not going to publish your paper.
Yeah, I think when we wrote that paper from a couple of years ago, we had kind of a suggestion that instead of people calling a GWAS when they were analyzing the autosomes, that they should only be allowed to call it an autosome-wide association study if they didn't analyze the sex chromosomes. But either within discussion between the authors on the paper and probably the reviewers, I think we ended up deciding removing that part from the paper. just because one of the reviewers thought that there was no need for any more acronyms.
Yeah. Dr. Paterson, thank you so much for all the work that you do and for this conversation. To put everything in perspective, all the things that we talked about, what does it mean for the average person?
So I think to put the perspective on genetic studies of complex traits and quantitative traits, we're still relatively early on. Like the first convincing genetic association studies were published less than 20 years ago. And it's only in the last 5 or 10 years that we've started to have really well-powered, large-scale genome-wide association studies of most of the important diseases and traits. And it's only now that people are beginning to be able to design studies where they can test whether including that information, the genetic information, potentially has beneficial impact for public health. So, for example, we can think of kind of conventional screening programs for cancer. So breast cancer, mammograms, colorectal cancer, kind of fecal occult blood tests. So kind of at the moment, most of these screening programs are provided to the population without incorporating any known risk factors. If we could make these programs more efficient, such that the people who are at highest risk have the highest intensity of screening, and the people that are at lowest risk have the lowest intensity of screening, or in some cases perhaps even no screening, then that potentially could be a public health improvement. It potentially could save money and could potentially have clinical benefits. So trying to incorporate genetics into routine medical care is still a big challenge. Like, we first have to make sure that we're not doing harm, and then we have to see whether we can actually do some benefit with it. But there are large-scale studies attempting to address both of these questions at the moment.
Amazing. And let's end it with this one. What advice do you have for me as a PhD student?
I mean, one of the advantages that I've had throughout my research career is there have always been interesting questions to ask. And coming into work every day has never been a challenge of motivation. There's always been lots of questions that we could ask and the resources to be able to answer at least some of them or make progress on them. So kind of making sure that the thing that you're doing is likely to have some positive impact on somebody in the future. I mean, knowing whether that's really going to be the case with every single action is a bit challenging, but kind of having that viewpoint, I think, has helped me.
Great. Thank you so much. I think it was amazing.
Thank you. It's pretty warm in here.
I know. We can't have AC because of the audio and everything.
Yeah, the noise. Yeah.